My name is Naar. I'm a professor at the University of California in Berkeley in Metabolic Biology. Just moved from Harvard Medical School, where I've been a professor for 17 years, and also the Massachusetts General Hospital Cancer Center, where I was a PI. We've been studying cholesterol lipid regulation for many years, trying to understand the molecular mechanism by which transcription factors and microRNAs control cholesterol and lipids. And more recently, we have stumbled on a microRNA that appears to be a crucial cholesterol regulator, both in atherosclerosis and, as I presented at this meeting, in AMD. As a summary of the talk, I talked about the initial work that we've done on this microRNA called MER33 and its role in controlling cholesterol-lipid homeostasis in mouse models and in non-human primates, showing that this microRNA plays a crucial role in controlling HDL as well as cholesterol efflux from atherogenic macrophages, and has actually a pro-atherogenic role in atherogenic macrophages causing heart disease, and we can antagonize the microRNA with potent antisense technologies. I shifted then gear to the relevance of this pathway in AMD. AMD is characterized by cholesterol accumulation in the retina in areas called drusen. These are cholesterol and lipid-rich particles, if you will, in the retina. And we have been studying the role of this microRNA in the accumulation of cholesterol in the drusen. And interestingly, we found that the microRNA MER33 does control cholesterol in retinal pigment epithelial cells by virtue of controlling a drug efflux pump, or rather the cholesterol efflux pump ABCA1. ABCA1 is a crucial cholesterol efflux pump that contributes to decreasing atherosclerosis and decreasing cholesterol accumulation also in the eye. We also showed that the microRNA appears to control inflammation in the eye, and we showed that antagonism in mouse models, mice fed a Western type diet, we have quite profound effects on cholesterol accumulation in the eye, as well as inflammation and retinal pigment epithelial morphology. So it looked from the mouse studies fed a Western type diet that there was a very strong effect on AMD-like pathology. We then switched to a non-human primate model where we're studying the injection of the antisense into non-human primates, also fed a Western type diet. And there again, basically mimicking what happened in the mouse models, we see very strong effects on HDL cholesterol circulation. We also found strong efflux, increased efflux capacity in the RPs, increased ABCA1. There was also decreased inflammation in these in the RPs, and also increased or improved morphology of the RPs in the non-human primates. So we think that antisense targeting of MER33 might actually be an interesting therapeutic approach to AMD, as we validated this in 2 different animal models in both mice and non-human primates fed a Western type diet. So I think the future studies will then be to do IND-enabling toxicology studies in rodents and non-human primates, and then phase 1 in humans to look at the safety of anti-MER33 treatment, and then followed by phase 2 for AMD.
Have any of the current cholesterol medications been shown to be beneficial for treating AMD?
So I should have mentioned that there is some previous data with statin treatment, which is a cholesterol-lowering drug, that there may be, at least in a subset of patients, a decreased cholesterol accumulation in the eye and in the drusen in particular. So there was also data suggesting that in human patients there may be benefit of lowering cholesterol in the retina. I should also mention a prior study by a different group, Rajendra Apte at Washington University, had also shown that antisense targeting of MER33 could also impact the choroid neovascularization of CNV, which is a different form of AMD. It's a wet form that's more severe.
So targeting cholesterol may be effective in treating the early form or dry AMD?
believe that the dry form is probably primarily driven by the cholesterol accumulation, and whereas the wet form may be driven more by inflammatory processes.
How can these findings translate into patient care?
Well, again, this is very early stage animal research, and so that is unclear at this point. If we can find that this treatment is safe initially and then efficacious, that it's a long road to get to a treatment for patients. But perhaps, if everything goes well, it might be 5 to 10 years down the line.